Agriculture and Rural Development, Mr. Nguyễn Mạnh Cường, Ministry of Science and Technology, leader from Hùng Hậu Group, Dr. Từ Minh Thiện, President of Hien University, Dr. Hiroaki Funahashi from Okayama University, Dr. Kaylan Reyes from Ghent University, Professor Buchibu, keynote speaker, professor, participant, distinguished guests, ladies and gentlemen. Because of the chairman, Professor Thiện, already mentioned very detailed in his speech, so I'm briefly have only two ideas. At first, I think we all know that the fourth industrial revolution, called 4IR, is going on: automation, traditional manufacturing, and industrial practicing using modern smart technology. And now goes quickly to any fields of our life. The 4IR is starting to change how many agricultural players, from the family farmer to a global, produce and relate products. The threat of so-called essential aid technology, such as artificial intelligence, AI, blockchain, drone, and the Internet thing, IoT, to our agriculture, aim to increase and lead in the increase in yields, low costs, and reduce environmental impact. The tools are also empowering farms to unlock new plant-based innovation and increasing their resilience to the extreme weather and climate change. This morning, we are very happy that all the people gathering face-to-face here at the Hien University, and also online with Okayama University and Ghent University as well. So our international symposium already mentioned very clear in the brochure, so you can find out more. But on the behalf of member of organizing committee and also on the behalf of Tho University delegates, I wish that during a very short time of today, we will share all the information and we get know together more and more. And I hope that we can set up a networking during the symposium today for the future collaboration. And also myself, we appreciate and thank to Hien University for your kind support. You do extremely and excellent logistic preparation, and we have the symposium today. And also we appreciate for all the guests, participants, you share your busy time to come here. Even we are now facing with the coronavirus pandemic, but we are here today. Lastly, myself and our member from Tho University, we thank to Hien again, and we hope all of us here have a time to enjoy the beautiful campus today. Thank you.
Thank you, Dr. Từ Minh Thiện and Dr. Lê Việt Dũng for the informative opening speech. It's short, but provide us with a lot of information from apply agriculture, climate change, and apply technology in agriculture. Could you give a big hand to Dr. Thiện and Dr. Lê Việt Dũng for that excellent opening speech? We are a little bit ahead of time, so we are now continuing with our meeting to the next agenda. That is to hear the keynote speech, which will be delivered by Dr. Tho Minh Thiện. Dr. Thiện had been going through many positions prior joining Hien University as the executive president. Dr. Thiện was the director of Ho Chi Minh City Trade and Investment Promotion Center. He also the former member of Ho Chi Minh City People Council at the term 2011 and 2016. He also a vice president of Ho Chi Minh City Agriculture High-Tech Park. Dr. Thiện had been active in the field of high-tech agriculture for more than 10 years. He has been successful in calling on many international investment in high-tech agriculture in Ho Chi Minh City. Now, please welcome again Dr. Tho Minh Thiện for his keynote speech. Please welcome. Yeah, please keep it for 20 minutes.
Thank you, Dr. Quân. I would like to present the presentation. Please. Show it. Today, we will present high-tech application view of the crops in agriculture in the Mekong Delta area. Yeah. Sorry. Yeah. In the content, we have three parts. The first, we would like to introduce an overview of the Mekong Delta area, and the second is the overview of high-tech application in cultivation and in agriculture in general in the Mekong Delta. And the third is the firm suggestion on solution to promote to apply the high-tech in agriculture in the Mekong Delta area. You know, in Vietnam, you can see, we have 8 agricultural production zone in Vietnam. In the Mekong Delta, it accounted for the 34.1%. And the Mekong Delta is one of the 4 key economic regions of the Vietnam. And the natural area about 4 million hectares. In that, they provide more than 50% of rice production, and more than 30% of fruit, and 90% of rice export, and 60% of the seafood export turnover in the whole of country. So you can see in the very important role of the Mekong Delta region in the Vietnam in general. And beside that, in the Mekong Delta area, we have 1,800 agricultural cooperatives. It includes about 200,030 members, account for the 30% of the country. Yeah, it's very big. Next one, please. Okay. Now, you know, in Vietnam nowadays we are facing with 3 challenges. It's very hard for Vietnam to solve it. The first is climate change. You know, the climate change affects strongly in Vietnam, especially in the Mekong Delta area, about the drought, about the flood, about the immersion, about the salt vegetation. And the second one is industrial revolution 4.0 that the Professor Lê Việt Dũng said. Yeah, it's creates many chances for Vietnam, but beside that, it's very difficult for Vietnam. Very many challenges in Vietnam because we have to follow, we have to comply the international rule, international resolution. So it's very important, very hard for us to set up and to organize in the raw material area to accompany and to follow to the international rules. And the third one is international integration. With Vietnam to integrate at least 16 FTA, the free trade agreement. So we have to follow and we have to comply with the many international law and international regulation. And besides that, the last month, we just implement the EVFTA, the Vietnam EU, the free trade agreement. Yeah, it's open many opportunities for Vietnam, especially in agriculture, but also to set Vietnam facing many challenges, many difficulties to organizing the supply chain to suitable to the law of the international line. The second one I would like to introduce the overview of a high-tech application is the cultivation of Mekong Delta region. You know, in the cultivation, we apply the biotechnology in biological production for agriculture and environment with many microorganisms, algae, plants, fungi, etc., and a multiplication of technology using tissue culture and the cultivation without soil. So, for example, the hydroponics depends on floating bore technology and aeroponics and germination. Besides that, we can also use the sprinkler and the drip irrigation with automatic and semi-automatic control system, and using a greenhouse, polyethylene film houses with automatic or semi-automatic control system, and a fertilizer. We are also to use nanotechnology, hydroapatite, and slow release fertilizer. And you know, can see some of the picture to illustrate, to demonstrate. So, for example, in hydroponics, I use hydroponics and the automatic dripping irrigation system. Now, in Vietnam, that's why of the advantage of this solar energy. But now some of the company they set up in the plant factory, to capture and to cultivate the sickling, the free disease sickling. Yeah, it's very good for us now to create now some company. They not only to grow the exotic vegetable, but also some the ginseng from the Korea. They would like to grow the ginseng about 2 years from the Korea. After the 2 months, after that they can sell. Yeah. You know, there's some the picture of the plant factory. In that you can see, yeah, it's a protect or a very good environment. In that you can harvest and eat immediately. Yeah. And besides that, we also to apply to the temporary immersion system. They call the TIS system. Yeah, to shorten the time, to save the time to grow from the tissue and the sickling. Yeah. Some the company they set up the container, you know, set up container with a LED light inside. Yeah, with about 4 or 5 layers, you can grow many kind of the vegetable, especially the exotic vegetable imported from the foreign. Please, next one. Yeah, you can see that now the automatic fertilizer is not enough far, strength with the producer nowadays. You can see they use all the automatic fertilizer. They combine with the fertilizer and the water to supply the plant, and they use the Lego hydroponics system. You know, the Lego hydroponic system you can change, and it's very mobile. It's very flexible to grow the vegetable, and also to import and to stimulate the farm bot. The farm bot is like the automatic. It's like a robot. They can grow, they can nourish, and they can take care the all of the plant that you raise. And home farm. The home farm now is more popular in the restaurant, especially in the high-class restaurant. Yeah, because it's you can look like the ornamental object, and beside that you can harvest that you serve in the eating. I would like to expand the animal husbandry. Yeah, in animal husbandry, they use embryo cutting technique and improving the intro fertilization and frozen embryo sperm technology. And they apply the modern husbandry with assembly line and automatic control system. And producing and using a biotechnological products with natural origins to limit the use of antibiotic and producing the vaccine. And producing the biologies, the output supporting metabolism, such as enzyme, phytase, cellulase, protease, to use fish effectively and limit environmental pollution. For example, you can see it's the case of the poultry and pig farming system of biological safety. Yeah, it's suitable to the regulation of the animal husbandry. And besides that, now the dairy cow case farm, they using the system to control individual dairy cow. Yeah, they have a sensor to take on the parameter from dairy cow to the software and to treat and to transfer on information to in the TMR. You know, TMR machine. TMR is the total mix ratio machine. Yeah, it mix on the feed and provide in the individual cattle. Yeah, it's very important. It's very effective. And the milking system. Yeah. And the aquaculture, they apply to biofloc. Biofloc now I think is very They begin the more popular in the Vietnam, especially in the along the sea and in the south of the Vietnam for water treatment and disease prevention. We also apply the genetic and cell technology in producing disease-free brick with a resilient producing unisex and treefold bricks, coloring pet fish, and producing applied equipment and tools for prompt diagnosis of aquaculture disease. You can see some of the model of in aquaculture. For example, they use aquaponics model is a combination between the planting and the fish. It's very suitable to some specific fish. And in the US, the Virginia, the FDA, the Food Truck Association, administration, they confirm, and then they confirm the model of the aquaponics model in the town, not only in the individual model. Yeah. And some the province has the seas. They use many kinds of the new method to raise. For example, they use the SDPE stream farming in the sea. Yeah, and they use nanopopol in the pool of the water, to raise the aquaculture, to raise the shrimp and fish. Yeah. And especially now, they will apply to the blockchain to traceability, to track the origin of products, not only in the vegetable but also in the animal products. For example, the pork, the pork, and the next will the fish, and some article. The blockchain applied in many kinds, many kind of the fields. But in Vietnam, we have suitable to at least the three items. For example, the check your supply chain, And the second is to protect intellectual property rights and to apply Internet of Things in the real. Yeah. For example, in the QR code, yeah, to track the origin of the pork in Vietnam nowadays. Besides that, we also to apply the ICT. Oh, ICT is very, very effective in management. Yeah, with ICT, we can track many. We can apply the ICT in market approach and value chain, apply ICT in boosting productivity of plant and animal, and apply ICT in risk management, and apply ICT in land management. Yeah, the last one is apply ICT improve the food safety and original tracking of agricultural products. For example, now they use a lot of the smart agri system. You know, can you use in the smartphone? You can control. You can you can active on the operation with operate the greenhouse, the cage, etc. You know, it apply in the animal husbandry, in the planting, and in aquaculture. For example, you know, they use in the ICT to control the use of land. Yeah, and beside that, we also to combine with the solar energy with the planting produce. Everyone to know the effective and very important to apply the high tech in agriculture, but also, we are facing many challenges, many difficulties. For example, the first is we unstable IT engineering infrastructure, especially in the far of the central. Yeah. The second one is the interest in coordination between the management and the administration with the farmer, with the producer, with the enterprise. The third one is the scale of production in Vietnam now still limited. The small and the dispersed production scale is very popular. So we have to together with them to set up in the big one, yeah, to apply it easier for the agriculture high tech. And this distributing database. Database in Vietnam is very a lot, but it's not concentrated. It's not concentrated in the one side, but distributed many places. And it's one problem when we wanted to apply the platform. We want to apply to the blockchain. We want to apply to the ICT in agriculture. And we also to limit the capital investment. You know, because if we wanted to apply high tech, we have to more the investment capital. So it's also one of difficulties. And the output uncertainty and price. Yeah, the price and consuming is always, and it's very, very hard to solve it. You know, we harvest a lot of the output. We can not meet at a good price. Yeah, the price decline. So now it's the reason that the farmer they are facing and they get many difficult for them. And some suggestion for solution to promote high technology application in farming in the Mekong Delta region. We have about the 6 suggestions. The first, the connecting high-tech agriculture, the connect in domestic with some institute or the university, like Tho University, like the institute, other institute, and the other international agency, for example, Okayama. Yeah. The second one, we would like to forming the agricultural products connecting chains. Yeah, would like to set up the supply chain. From the supply chain, we can control on the quality and the quantity of the product. We can provide the right product, the right time, and the right place. And we can apply the technology and post-harvest in ICT. You know, post-harvest is a problem now. Every year we lost at least about 25% of the output caused by the weakness of the post-harvest. So we have increased and we enhance the capacity of the post-harvest technology. And the fifth is the international cooperation. Yeah. With to expand the international cooperation, we can apply many the advanced technology, advanced technique in agriculture to increase the yield, to increase the quality, to increase the income of farmer. And the last one, to high-tech agriculture forum. We would like to set up some of the forum. From a forum, we can collect all the problems from the real, and we find the solution. Right? After that, we can get back to the farmer and to the enterprises. Yeah. And for example, using the model to apply the ICT in agriculture with the change, the supply chain. You know the supply chain inside. In outside, we have two more for the system. For example, the first is GIS visualization system. The second one is GIS is the production management system. The third one is the traceability integrated GIS to control in the supply chain. And the last one is agricultural market information system. Yeah. And besides that, we also wanted to cooperate. Want to cooperate with the Okayama, with the Tho University, for example. We can cooperate to apply the ICT, the AI, the platform in agriculture. We can use the laboratory. Yeah, you can get the standard appraisal. We can cooperate to train the expert, to train the students, and we can do the student exchange, and the consultancy, research collaboration, and expert exchange, and the technical infrastructure. I think this cooperation, and we can get the network like Dr. Viet Dũng say. Yeah, we can set up a network among the scientists, among the scientists with the management, the administration. Yeah, and the producer, we can transfer technology, we can research, and we can apply all the new technology, new advanced technique in the real. Yeah, that's it. Yeah, it's all the brief information that I would like to express this to you, to present to you in the overall of the high-tech agriculture in Mekong Delta area. Yeah, thank you very much. Yeah.
Thank you, Dr. Thiện, very much for your presentation. A little bit more information about Dr. Thiện. Dr. Thiện just become the member of the Hien University family just for 4 months. So with seeing the changing under his leadership here. His presentation open opportunity for cooperation between us, between Hien University, between Tho University and Okayama University, and also the cooperation between Vietnam and Japan. We see a lot of opportunity for the future cooperation. And for our next keynote speaker, I'd like to invite Professor Hiroaki Funahashi. I see you ready, right? Professor Funahaki is current vice president of Okayama University. He also the dean of Graduate School of Environment and Life Science, as well as a professor of the Assisted Reproductive Technology Center at Okayama University, Japan. His major in animal reproductive biotechnology. He has published 11 book chapters, 110 peer-reviewed international journals, and many other presentations at scientific meetings. He was awarded Shimomura Award for his research achievement in 1998. He also the co-editor of two international journals, Reproduction in Domestic Animals and Journal of Animal Science. Now, please welcome Professor Hiroyaki Funahashi to deliver his presentation from Japan through our online platform. Please welcome Dr. Hiroyaki.
Thank you, Chairman. Good morning, everyone. Before I start my presentation, I'd like, on behalf of Okayama University, as a co-sponsor of this symposium on application of advanced technologies in agriculture, I'd like to extend my greetings. Okayama University is a national university in Okayama City, located in the western part of Japan's main island between Osaka and Hiroshima cities. Our university is a comprehensive research university with 11 faculties and 8 graduate schools, and is engaged in the various education and research programs to achieve the Sustainable Development Goals, SDGs. We have a dependent exchange since we signed academic exchange agreement with Hien University, the organizer of this symposium, in December 2018. And also with Kanto University, the other co-sponsor of this symposium in September 2019, as well as with Guelph University in December 2019. As a person related to the conclusion of both exchange agreements, I'm very deeply moved and grateful to be able to hold this symposium at this time. As one of the fruit of this academic exchange, in particular, this year, COVID-19 has taken the world by storm, forcing us into a new lifestyle. Despite such difficulty, we were able to hold this symposium through the web. We'd like to express our deepest gratitude to all people who have contributed to the holding of this symposium. Global climate change, not just COVID-19, is forcing us to make many changes. However, agriculture, which is responsible for the food production necessary for human survival, must adapt quickly to the environment and continue to provide safe food in stable manners. As the title of this symposium suggested, new technology must open up this challenge. From Okayama University, totally 9 professors and 9 graduate students are participating in this symposium, and their recent research results will be presented. We hope that this symposium will not only deepen our friendship, but also be a fruitful symposium for both Japan and Vietnam with achieved discussion. Although this is on the web, please talk with many people and enjoy this symposium. Thank you for your attention. So I'd like to start my presentation. Just a minute. どこで選ぶの？あ、これ。あ、これね。Can you my slides? 見えてます？ Okay, so following a nice presentation by Dr. Lee Bien-dun, the chairman of AATA 2020, I'd like to talk about sustainable animal production in the current changeable world. As FAO has pointed out, food production appears to be insufficient for current food demand in the world, especially in many developing countries, because the amount of food production couldn't cover increasing the population in the world. In such kind of situation, food production stagnation cannot be tolerated, and constant effort are needed to improve its production efficiency. As you know, animal production is the largest user of land in the world. Through grazing and consumption of fodder and feed grain, the difference is significant as compared with plant production. However, meat production, especially poultry and pig meat production, is increasing year by year, mainly in Asian countries. So animal production in Asian country is the biggest in the world. Meat consumption has also increased significantly in Asian countries for 50 years. CO2 emissions by animal production is the largest in Asian country, including Southeast and Southeast Asia. Also, greenhouse gas emission is significantly increasing in electricity area, and the increase in agriculture is not big as compared with such human activity. Green gas emission for animal production is a majority in agriculture field. You know, there is a high correlation between GDP per capita and meat consumption because livestock products has been used as a fast food for workers who have flowed into urban area since the Industrial Revolution and have supported affluent lives as an energy source for economic growth in developed countries. Meat consumption per capita in Vietnam is a little bit higher than Japan, so you may have more possibility to develop very quickly. However, livestock production in Asia has plenty of rooms to improve the efficiency. Here, this graph is showing the efficiency of poultry yield kilogram per animal. The efficiencies in Asian and African countries seems to be much lower than the average of the efficiency in the world. The efficiency in milk production, as well as those in other animal production in Asian and African countries, are similar. Also, the amount of milk production is drastically increasing in Asian countries. Therefore, improvement in the quality and functionality of their livestock product, as well as their production efficiency, are required for reducing anthropogenic greenhouse gas emission and for realizing sustainable livestock production to achieve sustainable developmental goals. In the current world, highly infectious and serious disease for domestic animals, such as foot and mouth disease and highly pathogenic avian influenza, are causing great damage to livestock production. Recently, African swine fever as well as classical swine fever are serious in the world. One of 4 of world pig dead by African swine fever in 2019. It is necessary to assess and manage the risks against such animal infection and to control serious animal disease to minimize adverse effect. Furthermore, in the context of current pandemic of COVID-19 having a serious long-term impact on global logistics, food production, challenges may need to be discussed, not only on global scale, but also on a slightly narrower scale. So, in the current review, new technology to realize sustainable improvement of animal production with protecting the environment, including animal biotechnology, contributing to the genetic improvement and diversity maintaining and the domestic animal, etc., will be discussed. Okay, so firstly, I'd like to talk about our research farm. Its name is Tsudaka Farm in Okayama University. The farm has totally 31 hectares, including 12 hectare grassland. We are producing and breeding of Japanese black cattle Wagyu, about 70 to 80 head. However, in this farm, only 2 persons is working in this to operate. So because we are using a lot of IoT technology, also many big machines. The cattle has each cattle has a step counter sending information to the computer every hours. So when the cattle has estrus, we can detect those estrus automatically. Also, in the farms, we are using microbiological technologies to reduce the bad smell from odor. So we can produce meat, very nice meat. You can see here, the price is very expensive, but the taste is very good. So now, in my country, this technology is very common now. So each farm starts to introduce this kind of technologies to detect estrus. So also, Japanese government tried to integrate this system to calculate and manage the animal quality, animal breedings. Also, timed artificial insemination, it means we don't need to require estrus detection. Is already developed, and now also this one is very becoming common year by year. So this technology is not required estrus detection. So a farmer is easily can produce cows. Also, for the artificial insemination, sexing technology is applying now. So we can separate spermatozoa with Y and or X chromosome. The accuracy is more than 90%. So easily we can produce the cattle, male or female cattle, which we want. In my laboratory, I also try to improve the efficiency of artificial insemination. We focus on the polymorphonuclear leukocyte because after artificial insemination, this PMN is increased and the phagocytotic activity of PMN is increased to reduce the spermatozoa we inseminate. So finally, we found if we add the caffeine and calcium to the insemination solution, we can reduce the total number of PMN after artificial insemination. Also, we could improve the efficiency of artificial insemination, I mean the pregnancy and the following rate. Also, we know now, in the presence, when we do the artificial insemination, usually we use egg yolk to maintain the sperm quality during the freezing and the thawing procedure. However, the presence of egg yolk stimulates the phagocytotic activity of porcine PMN. But we know now the bovine is not. So this result may be able to explain about poor fertility following artificial insemination with frozen thawed bull semen as compared with bulls. Also for the animal breedings or embryo transfer and multiple ovulation technique is very useful. However, recently, over pickup method is becoming very common now. This technology is usually cattle is ovulated every 21 days, but the follicular development is we have several waves during estrous cycle. So if we can get those oocytes during the estrous cycle, more oocyte we can obtain the more oocyte. So people try to collect the oocyte through the estrous cycle, then try to make a fertilization in vitro to produce embryos in vitro. So by using these technologies, genetic improvement is very stimulated. So now some people start to use these technologies. This is data in the production of bovine embryos in Brazil. In Brazil, now is very embryo production is very common in vitro as well as compared with in vivo. So this technology is more easy to produce embryos and also very cheap. So, okay, to combine these technologies, maybe we can produce more embryos efficiently. Also, we may stimulate the breeding efficiency. Okay. However, in cattle, in the pig also, usually we are collecting the oocyte for in vitro production embryos, mainly from the middle-sized follicle. It means 3 to 6 millimeter in diameter, because the oocyte need to grow with follicle development. So now we know if we collect the oocyte from the smaller follicle, the meiotic and developmental competence is much lower as compared with the oocyte derived from the middle-sized follicle. So now we in our laboratories we challenge to improve the efficiency and the competencies for meiosis and early development. Now we know the secretions derived from the surrounding cumulus cell may help to improve the ability. Then one of my Vietnamese student found the factor is vascular endocellular growth factor, VEGF. So if we supplement this factor into in vitro maturation medium, we can improve significantly the developmental competence. We can improve the developmental competence of embryos derived from the small follicle. Okay, so also another Japanese research group has already succeeded to produce sperm and oocyte derived from the embryonic stem cell, so in vitro. So it means just by using culture technique, now we can produce oocyte also and spermatozoa. So after in vitro fertilization, we can produce the mice from this embryonic stem cell. Okay, so if we use these technologies, we can establish the in vitro breeding. It means now we improve the genetic qualities in domestic animals. We need produce the many cattle and piglets to assess the quality. However, now by using this culture systems, we can produce the sperm and oocyte. Then we combine those genome to make new embryos. So if we assess these embryos, genetic analysis may clear which combination is better. So without calf and piglet production, we can do the breedings in vitro. Also now, we have good technologies, genome editing technologies by using CRISPR-Cas9. So now we can introduce or break the genes where we want. So this slide showing in our laboratories, we could produce the genome editing mice. These mice, so we destroy SRY genes on Y chromosome. Okay, so these mice, we say sex reverse mice, has XY sex chromosome set. So it should be the male. However, this, if we destroy SRY gene by genome editing technologies, this mice has uterus and ovaries. Then in the ovaries, they can produce the oocyte. So just by using these technologies, we can modify everything. So combined with these genome editing technologies and in vitro breedings, now we can make a new breeding system to produce some new, more better animals. The Belgian blue, this is a mutation in myostatin. But that is a natural mutation. However, we can manipulate such kind of, we can produce such kind of mutation by using these technologies and this breeding system. Also, this genome editing technology can produce the disease resistance domestic animals. Now, if we edit the animals, so we may reduce a lot of serious disease. Okay, so in conclusion, basic and conventional animal biotechnology have developed to improve the efficiency and combine together with those technologies to expand the application to not only animal production. Furthermore, current new biotechnologies drastically make it possible to improve and create genetically domestic animals and quickly expand the utilization for sustainable animal production in the changeable world. To accelerate sustainable animal production, which is friendly with current environment, further development and integration of cut-edge animal biotechnologies may be required. So this is an acknowledgement. I'd like to thank you to the organized committee of this symposium on application of advanced technology in agriculture. Especially special thanks to Dr. Lai QUICKOM for introducing me in this session. Also, Dr. Le Viet-Dun, the chairman of AATA 2020. Thank you very much for your attention.
Thank you very much, Dr. Hiroaki Funahashi, for your informative and impressive presentation about the level of technology applied in agriculture in Japan. We are here in Vietnam. Recently, the Vietnamese media talking about the cow. Your presentation is come in the right time. Em nói là cái bài trình bày này lúc mình cũng đang thảo luận về bò Việt Nam. All right, we should have a presentation from Japan for the animal science and animal biotechnology. Now, to our next speaker, I'd like to introduce Professor Bùi Chí Biểu. Professor Biểu is currently the chairman of the State Council for Professorship in Agriculture and Forestry. He also is a leading scientist on dry genetics and breeding in Vietnam. Dr. Biểu was one of the first people to lay the foundation for the establishment of the Mekong Delta Rye Institute. His current research interests are rye breeding, molecular breeding, and quantitative genetics. He published a total of 310 peer-reviewed journals. He awarded Ho Chi Minh City Prize for its achievement and contribution to the rice agriculture development in Vietnam in 2000. He also honored the Sena Hira Award by the International Rice Institute for outstanding research in developing the diversification of rice varieties in Vietnam. Professor Bew, in Vietnamese we call Thầy, he also is a well-known teacher who had trained many generations of scientists, researchers, agriculture engineers for Vietnam. Now please welcome Professor Bùi Chí Bew to the podium to deliver your keynote speech. Welcome, Professor Bew.
Slide, please. Distinguished all participants, ladies and gentlemen. Slide, please. Okay. I appreciate very much and thank the organizing committee invite me to become the keynote speaker this morning here. I would like to introduce all of you about the Vietnam rice technology in post-NGS era, NGS with next generation sequencing. This because this is very important in everywhere in the world. They discuss about the next generation sequencing and application, but in Vietnam we have very few research, very few outputs due to the very limited research fund. And because I'm working on rice more than 40 years up to now. And the first lesson for my breeding lecture is breeding between art and science. Art and science. And some said that, oh, only it is due to the arts, not science. The farmer can do. The farmer can breed. The farmer can create a new variety. Do you believe? I think no. This due to the science. Today, very clear. Consumer, processor, exporter, and food industry will try what rice variety to grow. Not like this before, about 5 years ago in Vietnam. Today in Cần Thơ City or in any province in Mekong Delta, last year you obtained 10 million tons. This year you must 10.2, something like that. This means we manage our rice production based on the quantity, not quality. Today different. And then what we are doing now. Cái pointer nó nằm chỗ nào bấm không cái màu đỏ, cái màu nút giữa, nút dưới à. Today we no. We are in a commercial breeding. And I think we are forgetting science. We are forgetting because we just opened a very big road to convey our rice to Europe due to the FDA, new FDA. And then we are forgetting science. We are forgetting the breeding technology. We are forgetting the new technology applied to the breeding. And we don't have any research for breeding materials like the parents, something like that. So we think that this from Eddington, just the article just published in Crop Science, very important for us. And rice breeders need to adapt to more production-oriented molecular breeding. More molecular breeding pipeline driven by end user. Because the quality property by end user now require the outbidders to change their thinking, to change their strategy. And, it based on the advanced SNP array chip, or we call the next generation sequencing. Fortunately, new resources for SNP development is very, very well developed up to now. We have the three polars. It's an ORISA SNP 3 sequencing, RISE SNP consortium, and sequencing gene bank. Is three, like we call the three pillars for our molecular breeding. And after that, we develop. We also have in the world area also high solution genome-wide association study. Before we only study about the linkage genetics or linkage map. Today we can use the GWAT to develop the association mapping without the crossing, without the progenies. We can combine many accession in the gene bank. We can do, and this is the advance. And the most important tool is the high throughput genotyping. High throughput genotyping with you look the number of SNP is very, very big, very, very big. And sorry. Because after that we have from DDoC method by Sanger, very old time. In the world, we have the very big, very important history event, the sequencing, DNA sequencing. But today, they develop very fast. From DDoC to automatic sequencing and then next generation sequencing. With the high throughput sequencing like this, the event in the world now, DNA sequencing cost is plummeting now. Very, very cheap now, very cheap. Early 2000, about look the figure, $1 billion or some $100 million. Now today, whole genome roughly $1,000. And the how to put the sequencing, just we have two very well-known scientists in Cambridge University, Dr. Balasubramanian and Klineman. They created the so-called the Illumina sequencing. And then due to the bioinformatics advance. Bioinformatics one help us develop the high throughput sequencing, and we jump to the metagenomics and short read resequencing or long read, and especially 454 biosequencing, and then it's very, very important, very key tools for our research and apply in rice breeding now. Okay, next, what is the next generation sequencing? This is a new method for sequencing genome at high speed and low cost, high speed and low cost. And it's well known as second generation sequencing or third generation sequencing. But different, a little bit different for protocol also. So, but it's based on the bioinformatics. Without bioinformatics, we cannot do. And then it's for after whole genome sequencing, we look, it's very, the book is very thick like this. We cannot understand anything without the SNP. This marker, we call it SNP genotyping, SNP array, or biochip. They call it biochip, something like that. SNP genotyping, you know, due to the advance of annotation. Annotation created by some scientists come from Russia. They went to US in the early 1990s decade and create the so-called FGNet, the software, FGNet tool. And it's very important for annotation. And they call like a softberry, commercially softberry. If you open a softberry, you have. With this SNP genotyping is the measure of genetic variation of single nucleotide polymorphism between numbers of a species, including rice. And a SNP array is a type for DNA microarray. We call DNA microarray. We have like this one example, approximately about more than 40,000 probes to have us a visual score about the so-called the gene expression level with the different color from blue dark color to red to orange. Orange are high, the highest expression. This means the functional gene. Number one, we would like to introduce to you about the nutrition qualities, mean high quality rice. Is the top objective for a rice breeder now. You think that we have three problems. Number one is deficiency in micronutrient such as iron and zinc. Very important. Number two, this is deficient in amino acid and protein, mostly lysine and methionine in rice. And number three, this deficient in vitamins like folic acid. Did many articles in the world, and in Vietnam we only have for zinc and iron. Vietnam we focus the zinc and iron, and I think this is a new trend for rice quality. This we focus how to improve the iron in rice, and to do this, very famous scientist from Erie. He originated from India, Dr. Srinivasulu. The first time he identified the genetic variation on glycemic index resistant starch. We call the GI value, genetic index. Unfortunately, in Vietnam, you know, we have a survey with Dr. Madame Fitzgerald from Australia. She working in rice bran quality lab in Erie for a long time. We cooperate with her, and we survey from Red River Delta and to Mekong Delta about more than 300 samples. Unfortunately, Vietnamese rice variety contain the GI index about 60 to 70%. It's very high. So why? Vietnamese people are suffering the diabetics very high. And then our predicting we have to reduce the GI value about 40 to 50. And then the first man, he used the high density SNP genotyping to identify which the breeding materials to meet the demand of how to have the new high yielding variety with low GI. Yeah. And low GI, also another scientist, the article published in 2018 about the glycemic index like this in chromosome 6. In chromosome 6, remember, in chromosome 6, very important. And then, but in Vietnam, very few research on the GI. We pay more attention to the low and intermediate amylose content. And we forget about the relationship between the GI and high amylose content, rice. Very important, and I think Mr. Tung, remember this one. When we release the new variety with the high amylose, please accept, don't refuse. And Tập đoàn Lộc Trời, it means Lộc Trời Group, also recommended the new variety with a high GABA. GABA means gamma butyric acid. It's very important to adjust the hypertension. It means the high blood pressure patient. Because in Vietnam, high blood pressure patient too many, numerous now. This the target gene encode the protein CYP 78813 is very important. But the problem is we use the brown rice, not polished rice. If you polish on, no, because when you polish, you will remove the embryo. Embryo is very important. Embryo is very important to contain a lot of GABA. And in Japan, they have the rice breeding for giant embryo breeding. This means a very big embryo. For zinc and iron. In Mekong Delta Rice Research Institute, Dr. Trần Cúc Hòa, also she pay more attention to iron toxicity. But we fail because, due to the high phytic acid in rice, wheat, maize, something like that, so that the phytic acid of phytate, like to absorb, to inhibited the zinc and iron absorbed in intestine. And then the problem is we use the program we call the LPA mutant from Oryza refubogon and from Oryza sativa. And we successful in Oryza refubogon Vietnam. And then we can have the mutant like this. You look the standard very dark. This means low LPA gene, and it's the white is a lot of phytate, and some mutant is very dark, very good. Unfortunately, after 3 years, it lost because due to the gene repair mechanism, and then the mutant not stable. It is the recessive mutant located in chromosome 3 and chromosome 4. And then this we have to identify new one. And come to the beta carotene variety. It takes at least 20 years. Today, more than 30 years. From 1990s initiated until 2016, we have, but not released due to the critical regulation in the world. And then this very, very important for rice transformation. But unfortunately, it's not developed well. And this is the very hot topic now when we export rice to Europe. And our colleagues in the Agricultural Research Genetic Research Institute in the north, Dr. Quách Hữu Trung, he cooperated with our colleagues in French to resequence Tám Xoang Bắc Ninh and Tám Xoang Hải Hậu. He detected more than 40,000 polymorphism in the whole genome of aromatic landrace. And he also identified the mutant with the 8 deletion in chromosome 8, the gene, they have the exon number 7. And 7 hotspots, the region of Novo SNP and indels were found in both the genome, 2 varieties. It's a very important article you can shoot. It's very, 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 very good. You look the first round, it's the blue color. This is the total SNP. And the center is the unique indel number to associated to the aroma in Tám Xoang variety. And the new chain like this, before we said that this the recessive gene, we call a fractal gene located in chromosome 8, but today different. Today different. This the gene encoded the protein BET2. BET2 is the control the aroma in rice. And the predominant function of BET2 with the mutant at the 8pp indel, it's even 8pp indel in chromosome 8. And after that, they identified the one haplotype, they call it HAP6. HAP6 is very important. They also use the tool, they call a single KSAP SNP, unique, very unique to HAP6, and very useful for a rice breeder to identify the aromatic or non-aromatic progenies. Okay, this is just published in 2020. You can look, you can see the news in our website, aasvn.org, and then very new. And this also by our group to identify you look the blue color, we can genotyping by graphical genotyping, and we use X progeny with blue color. This the allele content of the gene we control the aroma, and then this is also very useful to use the color to identify the progeny in BC3F2, and we also use SNP identify the deletion in chromosome 8, cause a mutant. And we use the MS to identify the protein 2AP, it's mean aroma from jasmine 85 and OM 49200, the same. It means OM 49200 can be considered as a little aromatic variety. And come to the QTL integration from white rice and from lentils into the cultivars, into high-yielding variety. And my colleague, Michael Thompson, unfortunately now he moved from Erie to US. But when he worked in Erie, we cooperate with him to do the 6K SNP array for genetics and breeding application is very useful, because of this we call the cost-benefit sweet spot for routine genetic and breeding application. Very important to track. We can track the QTL in progression from donors to recurrence. It's very useful. We can identify the linkage drugs. This means the not good linkage. We can identify the target gene from white rice, Oryza refugon, Oryza rivera, Oryza officinalis into the cultivars. This is a very important tool, and this the the tracking QTL integration in chromosome 6 and 9. Chromosome 6 is include the XA7 gene control the Sanctomonas bacterial light. Very, very important in the north. And chromosome 9 is located sub1 gene, control the submersion tolerance. Submersion tolerance can control about 17 days, the maximum day to be submerged. It can recover. So this is the comparison, the so-called golden gate. Golden gate assay is a very strong tool now for genotyping platform and for genomic database. And then very important, we can apply in our reading program. And Thompson is very young scientist, and we have many outputs, very useful for rice breeder now in the world. We cooperate with Professor Ishikawa in Hirosaki, Japan, for P deficiency tolerance. In the world now, we face the problem for the P deficiency or P starvation due to the soil become more acidic due to climate change, like acidic soil in Mekong Delta. A lot of aluminum and iron, they fix the P, the phosphorus, cannot become available for rice to absorb. And we mentioned about, we use the method we call the map read and SNP detecting by alumina high sequence. And then this is published in the journal in America last year. And we use the AS996, the derivative from Horizon Nivara from Tam Chim, Dong Thap Mười, and IR64 with the total sequence like this. The blue color is the sequence by IR64, and the orange color is AS96 from white rice. This is the BUP1, the BUP1 in chromosome 12. And you look the BUP1 in chromosome 12, and it's not included in many promising progeny in Vietnam. This means in Vietnam, we don't have the BUP1 gene, but we have the STARK1, starvation P deficiency in chromosome 12. Next, we have one lady, Đinh Thị Lam. She working PhD with the chromosome 4. Chromosome 4 control the tubular capacity because the more tolerant, more tubular capacity, and it's located in chromosome 4 like this. Look in the whole 12 chromosome, and you look chromosome 4 and chromosome 12 with the more orange color. It means more SNP identify the target gene, target QTL. This based on the QTL map we mapped before, and then we also identify the target region in chromosome 12, at least about 1 or 2 megabase. And after that, we narrowing them, narrow down the area by SNP array. And we also use the CAPS, is mean the cleavage amplified polymorphism sign marker using the NAP cutter version 2.0 to identify the genotype. This is the parent, how to use the as a donor. Also, SNP array acid selection in chromosome 8 for this is very new research in the world, because in the world they pay more attention to the chromosome 12, but we also identify the new chromosome 8, 4 to have some gene like this to control the P deficiency, P starvation tolerance. And this one example of SNP array acid selection also. This is very useful in our breeding work. Based on the reference genome in rice, without the reference genome, we cannot do anything. Without bioinformatics, cannot. But fortunately, we have very good reference genome in rice, and we can apply the next generation in high throughput sequencing system to accomplish the whole genome resequencing in a very genetic population, and there was germplasm resource. So for Sonterran also, it's a very, very hot topic now in Mekong Delta. And then it depends on the two stages. Number one is the seedling stage. This is the seedling stage. Number two is the reproductive stage. And the tillering stage, no problem. But they strongly affected in seedling stage at the 3 leaves on to 6 leaves. And the reproductive is meant from the panicle initiation to flowering. And then before we only pay more attention to the chromosome 1. But now different. They use the haplotype. They identify the hap1 of the so-called OSSTL1, identify the they call it superior haplotype and non-synonymous SNP is most likely to be functional site. It's very important information for the rice breeder now. And then after that, we also, based on the GT map on two parent OM 1490 and Pokali. You look, the Pokali is the whole red, the blue color. Pokali is tolerant, and the red color is suitable. And then the progeny like this we can identify in chromosome 1 before. But today, not only in chromosome 1, many chromosomes up to the haplotype with the SNP array. This is the same we published last year or so. Then very difficult to identify the variety or genotype tolerant salinity in reproductive when flower. Oh, very difficult. But we use the association mapping GWAT to identify this. Our student he worked in Erie for master thesis. Identified for spikelet fertility percentage located in chromosome 8, like the same I'll conclude before. This used the 44,000 SNP chip to identify significant at the reproductive stage. Is the EC about 10 DS per meter. This only in the lab, not in the rice field. And the unfilled grand number panicle also located in the chromosome 4, and this the linkage map also meet the fit the association mapping. And the last one is the heat tolerance. Heat tolerance is very important. I also the project leader of heat tolerance in Ministry of Agriculture for at least 6 years, and we do this. But the new one is they identify the transcription factors in the signal transduction. They call the BZIP74. Very important for me. But before that, we identify only two chromosomes, 3 and 4. We call this the canonical method, and we use the high solution map like this. We identify new marker, but we also successful for identify the protein for Western blot. It's not the HSB70, but this in our case the HU protein 90 is very important. And but when we compare to the output from France, our colleagues, Dr. Lafarge, in plus one, published in the plus one, they identify more. We saw the trace in chromosome 5, 8, and 19, but today if she use the GWAS and haplotypes combined to SNP, they have very clear. But before that, for the canonical method, we cannot do like this. Okay. After that, we can conclude like this. Looking in the future is risky due to climate change, very important in Mekong Delta. Due to the energy efficiency, due to the financial crisis. Financial crisis today due to the COVID-19, very important. And breeding still considered as the heart of agriculture production improvement. Something we forget about this. We pay more attention to the commercial breeding. We don't pay more attention to the basic research. And lesson 3, how to shorten the process from our scientific information to farmer knowledge. How to shorten the basic research to applied research. And the last one is, without international cooperation, our successful very late. With the international cooperation, it will be to access our technology. Thank you very much for your listening.
Thank you very much, Dr. Bill, for your presentation. And we are happy to see your ongoing passion to research, to write research in Vietnam. And we wish you a good health so you can continue contributing to research and training in Vietnam. Could we give a big hand to Dr. Bill for his presentation? For our next keynote speaker, I would like to invite Professor Kalen Rees. Professor Rees is the head of research unit VEZ in the Department of Food Technology, Safety and Health at Hien University, which is focused on 2 research lines: fermentation and chemical and microbiological quality. Professor Rees had 130 international peer-reviewed journals and more than 3,000 citations. Now, please welcome Professor Kalen Rees to deliver the keynote speech. Her presentation will from Belgium via our online platform. Please welcome Dr. Kalen Rees.
Hello everyone. First of all, I would like to thank the organization committee to invite me for giving a presentation. Unfortunately, I am not able to be with you in Vietnam, but I hope by this presentation and by the online systems, you can at least get some ideas from my presentation if needed. You can always contact me, of course. At the end of the presentation, you will find my contact details. So I'm Kathleen Razen from Hien University, and one of the research lines that's going on in my lab is regarding food waste valorization. Before starting with some examples that we have done in our lab, I found a very recent paper that focuses on the terminology that we are using at the moment for indicating, in fact, all the same things: food loss, food waste, surplus food, food byproducts, food side streams. All these terms are used and indicating that we have an amount of food that is or not used, or an amount of food that in fact is there as a byproduct because it's just not edible for the consumer. So this recent paper indicated a definition for food loss, which is an unintentional decrease in edible food, mainly due to post-harvest losses, but also processing losses. Food that is damaged due to transportation during the logistics. While food waste is most often used in the primary retail, so parts of the food that is not able to be consumed by people. We have these different categories of food: surplus food, food waste, and parts of it that are not edible at all for the consumer, as well as food losses. You see a whole cascade reaction where, of course, surplus food is needs to be prevented so that we don't overproduce and that we don't lose a lot of energy, a lot of nutrient sources. But in the food waste sector, or in the food waste category, there is a high priority for biorefining. So there are a lot of molecules, a lot of components present that we can try to reuse in food, in feed, in materials for agriculture and for energy. And of course, we always try to first valorize it in the high added value applications and later on in lower added value applications. However, in this biorefinery, attention has also to be paid to a zero waste approach so that we can valorize every fraction that we obtain and not that we valorize one specific compound and in the meantime producing another waste stream that has low value because of solvents that were used or that has to be burned because it becomes too toxic. So in most cases, you are now really trying to valorize all parts in the edible food waste approach. There are different types of valorization routes, and this is a nice example of different waste streams that has been tested, which are marked here on the left side of the circle, going from plant-based products, from vegetables, oilseed crops, fruits, cereals, to the animal-based products like dairy products, fish-based and meat-based products. And the extent of the colored part here indicates the number of researches that has been done on this type of food waste, but also indicates on the other side which added value products that has been obtained from these food waste products, and that has found an application in different sectors, in the agriculture, in the medical industry, and the food industry. You see that most of the work has been done on fruits, going for polysaccharides, mainly pectins, chitin, chitin, but also a large part went to the polyphenol research and applications and to bioactive compounds like carotenoids like bean and others. You can see that less value-added products has been found in vegetables in certain application. For cereals, we have mostly focused on added values in enzymes. So this is a very nice overview on all the type of food waste with a different approach in value-added products with a clear application in research. So it's not we find separate type of phenolics, but we find these phenolics in the fruit waste stream, and we also try it and have an application in bread and an application in a capsule as a food supplement. So this is a nice overview of all type of valorization routes that can going on and that can find its application in the industry. Knowing this, I will go with you through 3 examples of valorization of food waste products that have been done in our lab. The first one is based on fruit byproducts with a specific aim to extract and to valorize phenolic compounds. This is in fact a PhD of Phuong Nhat Nguyen that has been done in collaboration with Tho University and supported by the US of Belgium. In that PhD, we have focused on different types of Southeast Asian fruit waste products, and because mainly they cause environmental problems. You produce a large amount of fruits, a large amount of fruit waste are generated, but these byproducts still have added value, but at the moment almost nothing is done with it, and they are just stayed there, meaning that they start to rot and cause environmental problems and of course also economic losses. So what we try to do is to extract specifically phenolic compounds. These extracts, we try them, and after testing, we try to apply them as an antioxidant, because we saw that some of them has a very good antioxidant capacity. This in cold oil storage and in frying oil experiments. We also did some antimicrobial activity testing, not only in vitro, but also in situ on meat and fish. And we also screened for enzyme inhibitory properties. I will just talk on the application of the antioxidant activity, but of course more of the information can be found in the published papers and in the submit or in the defended PhD. After screening different types of fruit byproducts from Southeast Asia, we have chosen to work further with rambutan fruits, because rambutan fruits, as you see here in this scheme, produce more than 60% of food waste. So inedible parts by peel and by seed, and less than 40% is really edible for humans. So this means, as mentioned here, if we produce 100,000 tons of rambutan, that 63,000 tons of byproduct seeds and peels are produced. Of course, as you can see on the color, we can expect that there are some interesting compounds as well in these peels. We focused in this PhD on phenolic compounds, and after a first screening and identification of all the phenolic compounds that were present, we detected more than 40 different phenolic compounds in rambutan peel. The major ones that we found were ellagic acids, quercetin and its glucosides, and then very special phenolic compounds, geranium and corylagine. These are typical phenolic compounds belonging to rambutan and some associated fruits. Knowing that these rambutan peels were high in these 5 phenolic compounds, knowing that phenolic compounds has an antioxidant capacity, and also by the in vitro test, we confirmed that our extract has a high antioxidative capacity. We were able to test the antioxidant activity in soybean oil to have a further application. So therefore, we extracted rambutan peels with methanol, but also with water. After extraction, we dried the extract so that we received a powder and rambutan peel extract. And that rambutan peel extract was supplemented to, on the one hand, soybean oil that has underwent cold storage, or we added in soybean oil that would aim to fry food products, and in our case, we choose for potatoes. We measured different types of parameters, peroxide value, T-bars value, for estimating the oxidation potential. And we also did some quality check on the oil and the fried potatoes as well by measuring color. In both cases, we used a blank without any antioxidants added, so without our rambutan peel extract. And we also used a positive control, the TBHQ, as it's commonly used as a synthetic antioxidant in oil. Our rambutan peel extract was supplemented to the different oils, and the amount that we supplemented was based on the total phenolic compounds content that we obtained by the Foley method. If we look to the antioxidative stability or the antioxidant activity in soybean oil during cold storage, so we stored it at 30 degrees, or under light, or in the dark. Then we see two phenomena. In light, we don't see any or almost any difference between the different rambutan peel extracts compared to the control. So there is no inhibition of lipid oxidation, or there is no larger lag phase before oxidation is starting. So in two cases it's higher, and in one case it's lower than the control. So we don't see any effect of our rambutan peel. However, when we look to the dark storage of the soybean oils, then we see that the lag phase is increased, so we retard lipid oxidation during storage. We see with the blue line with control without any antioxidants already an increase in peroxide value from 40 days onwards, while this is not the case for the rambutan peel extract supplemented soybean oils. However, it cannot stabilize the oil in the same way as TBHQ. So after day 80, we see a clear increase in peroxide value, an increase that is higher than the limit that is given for good quality oil. When we look then to the antioxidative potential of these rambutan peel extracts in a heated model system, so when we add the rambutan peel extracts to the soybean oil for frying, then we measured the secondary oxidation products of the oil itself as well as of the potatoes. And we see that we have, in fact, a clear increase in the TBR's value by the control. But interestingly, we see no difference at all between our rambutan peel extract and our positive control, the TBHQ. And a similar effect we noticed in the potato, so where we fried 20 different batches of potatoes. Even after 20 batches of frying, we don't see any effects of the rambutan peel that was different compared to the positive control. But we see a clear increase in TBHQ for the control, so without any antioxidants. So we were able, with the rambutan peel extract, to have an activity, an antioxidant activity that was similar to the activity of TBHQ, only in a heated soybean oil model system. Of oil, that we can have degradation products of these phenolic compounds, because we all know that phenolic compounds are not heat stable, and that we have to study more in detail if there are any toxic byproducts formed or not during frying of a soybean oil. But also cereal brands, potato, steam peels, all these kind of byproducts are generated, but we all know that they are very high in fibers. At this moment, there is almost no applications done about the fibers. So what can we do with these fibers, and can we reuse it as a carrier, in this case for probiotics, or can we reuse it for something else? So the aim of this study was first to extract from all these agricultural byproducts the alcohol insoluble residue. So that means alcohol insoluble residue, so the fiber residue that we obtained was tested on the one hand for their antioxidant capacity, because we also know that on the fibers, if you look to the cell matrix, on the fibers itself, we still have phenolic compounds present that are really bound very closely to the fiber fraction. So we did a long-term antioxidant capacity measurements up to 4 hours. And in this case, you see that we have like 2 clusters of products. On the one hand, we have here a cluster that has a very high long-term antioxidant capacity. So like very.
So this is a video. So we stop here and we continue to play it during our break. And I know everyone here is looking for the break, and please have break, and we come back here at 11, and we have it open for discussion. And if you have any question, please hold after break, or you can write it down and deliver to the presiding board, and they will deliver. They will facilitate the discussion section after break. Please come back here at 11.
Giờ nghe, tại vì em phải nhắc ít khi không đố giờ.
Dạ, không sao
Đố nhiều cũng không.
Dạ, cũng đố một chút xíu. Dạ, không sao.
Xin lỗi là làm nãy giờ nó kẹt đi ra thì sao?
I would like to invite everyone to come up here for the Xin mời các thầy cô lên đây để mình chụp hình kỷ niệm ạ. Chụp kiểu gì? Em xin mời các thầy cô mình bước lên trên sân khấu để mình chụp hình kỷ niệm ạ. Xin mời các thầy cô. Dạ. Cái này là kéo cái này lên.
presentation is smart agriculture. Smart agriculture is a new type of agriculture that uses robots and IoT and ICT technologies to improve productivity while maintaining
Ladies and gentlemen, I hope you enjoy break and you had a little bit time to digest on a lot of information from 4 presentation in the morning. Now I would like to open floor for discussion. Dr. Hiroaki, are you ready from your side for the discussion? Uyên ơi, hình như cái bên Nhật người ta chưa không có ở đấy hả? Có không? Sao không thấy hình người ta? Okay. Em nhắn tin đi, Uyên nhắn tin mà người ta chuẩn bị để thảo luận đi. Dr. Zhu, are you there? Tắt camera. Em bảo họ bật camera lên đi. Okay, good. I can see you now. So, are you ready from Japan for the discussion?
Yes, we are ready.
Please raise your hand if you are ready. Okay, good. Okay, good. So now I would like to open floor for the discussion. I will hand my microphone over to our presiding board. And if you have any question, please raise your hand, or you can write down your question and give it to the facilitator who will facilitate the discussion today. I would like to invite Dr. Từ Minh Thiện, Dr. Lê Việt Dũng, and Dr. Đinh Thị Thủy will facilitate this section. Please.
Well, thank you, Mr. Facilitator. Now it's an open discussion, but let me have a very brief summary that the 4 keynotes present this morning. So we are very happy, and it's great that the topic of 4 keynotes is very science, deeply in different disciplines. At the beginning, Professor Hien gave the idea and some discussion about smart agriculture. And he mentions a lot about supply chains, value chains, and also he raised the new idea for crop production in terms of plant factory or fishery in bioblock. Secondly, Professor Hiroaki Funahashi shift to other topics related to the animal science. And also he mentions a lot hot issue that also relate to Vietnam, such like African swine fever, mouth foot disease. The third one, we listen deeply from Professor Bưu, who is extremely VIP expert in rice genetics, not only to Vietnam, but also for the international. And Professor Bưu let us know updated, latest advanced technology in DNA for rice genetic and reading. And we all know that he raised the term of NGS, the next generation of sequencing. Lastly, we listened to Professor Kalen raised in Ghent University of Belgium. So Professor Kalen invited to know more about the food and valorization rules and also the products of fruit. So I think 4 topics completely represent for the big major in our situation right now, not only in Vietnam, but also in Japan and again, Belgium as well. So now there's an open discussion. At first, we would like to invite all the participants who have the question or sharing or discussion following to the 4 topics of the 4 keynotes, please. Maybe first, Professor Cheung. Professor is the Vice Director of the Biotech Research Institute of Tho University. Professor Cheung, please.
Okay, thank you very much, organizer. Give me a chance to share my idea. So first of all, I think this is very interesting symposium. We in the keynote speech we can hear some information about high technology in agriculture. So Dr. Thiện, he summarized and give me a lot of interesting idea. And so in our university, around more than 45 years ago, we also pay attention on high technology to apply for plant tissue culture or hydroponic culture. But sometimes we think about the problem when we apply high technology. Sometimes we also apply some kind of chemical. For example, in my field, we research about plant growth substances. So now I don't want to call plant growth hormone or plant growth regulator. We call plant growth substances to emphasize on the substances we can apply for plant, not only plant hormone or phytohormone. But when we also get the problem when we apply high technology, we push our right to get the high yield for tropical root, also high yield. But now not only high yield but high quality and safety. Another problem, our agriculture product very cheap. Sometimes high yield but rice very cheap. So I remember that around 17 years ago in Tho University, we have the farmer to increase rambutan yield because sometimes they get the problem with the fertilizer. So we have the farmer increase rambutan yield. And that time, the yield of rambutan took 3,000 đồng per kilo. So when the project is success, we got a lot of rambutan. So the price jumped down just 3,000 đồng per kilo. So we must think another way how to rotate our product. So now a lot of research, also interesting research on root. And Professor Funahashi give me a picture about the animal production science. It's basically apply a lot of biotechnology technique for animal production. So it make me to remind about Tho University in the past. We also pay attention a lot of research. Or we also have the not department that time we have the faculty of animal husbandry and veterinary. But now today we have a lot of research about plant production. But I think not only plant production, we should push on a plant production in animal production in our reason because animal products give the high value compared with plant product. So when we take a break, I have a short question or short discussion with Professor Bố. I said that so in excellent his lecture make me remind a lot of high techniques, and I wake up and I would like to continue to study about genetics, because when I was a student I also have the graduation thesis with genetics, and Professor Bố gave me a lot of information and review some biotechnique, but I would like to have a question because in the past I also have a short time to research about rice lodging. Yeah, that time we use some kind of plant growth substances as the anti-gibberellin to control rice plant height. But talking about the biotechnology, or we can, is any information about the gene control the rice plants or control rice lodging, for example, can control about the cellulose or something in the membranes or in the wall. Yeah, if you have some information about that, we explain to it. We would like to apply some biotechnology to detect the gene in our gene bank to collect about some gene or some characteristics about rice lodging resistance or tolerance. Yeah. Finally, we would like to thank the report of Professor Caroline Ray. In our institute, we also have a lot of similar research about the antioxidant or try to detect some biological compounds in plant, in microorganism, but we lack a facility. We could not do our research at the end, try to find and recognize some active compound in our product. We also do something with rambutan or mangosteen or organism, but I hope in the future we can enlarge our research follow her lecture. Yeah, thank you very much. And that's our idea, and I would like to share to our symposium. Yeah, thank you.
Thank you, Dr. Chung. So may we recommend that, because now we have only 2 locations in Văn Hiến here of Vietnam and Okayama University, because Ghent University now is too late. Therefore, we only have 2 sites. That's the reason why we suggest the question we raise forward to Okayama only. And then from Okayama, we ready to listen to your question or discussion. So that's why we can ask you. For the Vietnamese here, we can discuss later on. So if you are agree, we start. How about Okayama University? So please send your question to our side, please.
Hello everyone. Can you hear me? Okay. It's in Japan now is 1:15. So thanks for your kindness. We are very happiness to attend the symposium. So, but sorry. For keynote speakers, speech presentation is a very good presentation, but my specialty is botanical, so I cannot understand. I cannot have any question. Sorry. Okay, so and we keep good connection for between Okayama University and Hien University and Kanto University. That's a great good situation. So, that's all. Thanks.
In our side, Professor Wu, please.
Hello.
Yes, it's a very interesting question on logging tolerance, because logging tolerance are related to high plants, high reduction, related to the canopy, related to the grand filling stage, very important to reduce the yield. And so far, we have many articles to report about the genetic background of logging tolerance. Number one is they identify already the QTLs to control the COM1 thickness, the COM diameters, the particle weight, and the problem is they cannot explain about the grand fueling, now, and then because first I think you know where they identified the dwarf gene very about more 40 years ago. Dwarf gene, they reduced the plant height, but today they identify more QTLs. But because the time not enough to explain to you about something, but they have today have identified due to the morphological, physiological study, and then genetics also. Yeah, a lot of reports like this. Thank you.
Thank you, Professor Wu. So with Okayama University, you have any further questions?
Okay, so now we are asking that for student. Hi, Japanese Okayama University student. Do you have any questions? If you have a question, please ask. Please.
If you don't feel too hungry, please ask question. And after that, we have break. Okay, we had that one hand raised over there.
Yes, your shell introduction, please.
My name is Ngân. I'm an associate professor from the College of Environment and Natural Resources of Tho University. So I think now is you will mention, because I just said that I'm from the environment. So I have one question first for Professor, the president of Hien University, because when you mentioned about the advanced technology for agriculture, you mentioned about the blockchain. I'm appreciate for that. I'm totally agree. That should be the future for agriculture activity. However, from my point of view, I found that the environment issues is not yet clearly mentioned from your presentation. And I recommend it could be some environment issue should be added in the blockchain. Because on you may know, in the case of business, if you pay more attention in environment, I'm sure you get the less benefit, less income. So if we don't put in advance the environmental issues in the process, so later on we don't want to put, and we cannot save for the future. Yeah, I also have to like one issues because I'm working in the field of apply the residue from agriculture activity to turn to energy. Yeah, so I learned that like the residue from agriculture, that is not the waste. That's still using the input for the energy production. So if we think in advance, we can integrate on the environment issues, so we can have the very well-known process for advanced technology for agriculture. Thank you.
Good morning. My name is Lý Nguyễn Bình from the College of Agriculture, Tho University. My background about food science and technology, but I would like to raise a question to Rosa Bu about the rice variety. Some years ago, I read one of your paper talking about the low GI rice variety, and you suggest that we should think about a kind of low GI rice variety in the future for not only Vietnam but also for the consumption in the world. I have no information and background about the genetic technology, but I know that our government, I mean Vietnamese government and also Japanese government, not allow to do the genetic modification of rice. So can you comment a little bit about how to make a new rice variety in terms of low GI? Thank you.
Yes. You know, who is the first suggest the low GI genotype? This is due to the Kellogg group in the USA, you know, very famous. When they cooperate with us during the 2010. And then I identified that they pay more attention to the low glycemic index. And then I don't know why, because they said that they didn't answer. And after that, we cooperate with the ERI scientists. And then we understand that this is a very important trait. We can improve it by conventional breeding. No need the GMO, no need the GM. And then the gene encode the protein, located in the chromosome 6. And we have many parents, each donors, to give the gene. But the problem is they have what we call the linkage drug. Linkage drug means the linkage with the very poor economic character. And then we have to disrupt the linkage drug like this. and then it's very easy. No, not but we need the money to to do this, because they identify due to the high SNP quality data from Erie we can use, no problem.
Yes, I just have a small question. So maybe we should think about a new project dealing with the low GI rice variety in the future between Tho University, Hien University, and Okayama University about that, because that low GI rice variety is a new trend for the world, because we have problem with diabetes. Thank you.
So, I am.
Okay.
Hello, I am Đinh Thị Thủy from Hien University. I have the question relationship about the shark catfish in the Mekong Delta, because I met the project increase the shark survival. The shark catfish very important in the Mekong Delta, Vietnam also. We can use this, but we have problem. We apply many new methods about increasing the survival and feeding the correct fish, and we did the new methods for fish management and disease. We have problem about disease prevention and treatment. In the Research Institute for Aquaculture #2, we have many projects about try to do, but we finally have the fish productivity is not so very high. In this in here, I want to every people, the scientists here talk about the how to use to apply the high technology in here for increasing the catfish in the Mekong Delta. Cô Phượng, mời cô Phượng ạ.
Hello, everybody. So I also have two questions. But the first question I would like to tell. Yeah. The first question I would like to ask Professor Thị. You talk everything about the advantage for in agriculture, but everything advantage and disadvantage. But in Vietnam, at present and in the future, what will you do? I want to ask you this. Maybe near future for 5 years, just not the next 5 years, just not so much so far. But besides that, I also have another question to Professor Bùi Chí Bụ. We don't know anything more about the genetic on rice, and so this is very complicated. But I know about some kind of rice, especially in Vietnam now. The AT25, AT24, this is the best rice in the world. They talk about it, but I don't think it's actually. But for how to produce this kind of rice, it's only produced by the engineering. It's not some scientists, but maybe maybe you can supply nutrients, supply the method for planting, supply some organic fertilizer, something to produce the best quality for the rice. According to your experience, you can explain us more detail about it, please. Thank you.
Please, first question go to Dr. Thiện, and second question go to Dr. Bew, please.
Yeah, thank you for your question. I would like to answer your question first. Yeah, I agree with you. If you pay more environment, you will get less profit in our day. Yeah, but I think a recycle is a good way for agriculture. The problem is now we have to combine to combine with the benefit from the environment and the production. I give you some example. You know, when I go to American, I deal with in the American company, very high technology. Yeah, they process from the rice husk into the semiconductor, RFID. I discussed with them, in the Mekong, some province, and I work with the Đồng Tháp province, An Giang province about that, and they agree to set up the milk, to set up the plant and the factory to collect all the rice husks from Mekong Delta to process in the RFID and the semiconductor or the toothpaste, and depend on the filter. Discriminate for an organic product or in the standard product. You know, because if you apply the chemical, you can get the high yield. Yeah, but it can affect and can harm to the user and consumer. And also we have to find the new distribution channel for them. We have to find the new in the niche market for them. I did that many years in Ho Chi Minh City. Yeah, I help many enterprise, many company to set up in the supply chain and to set up the connection between the raw material area and in the distribution channel. For example, the Lotte on the Saigon Cup market. Yeah, and I don't remember exactly the name, for example. But I think if we would like to pay the environment, we have to find a good way to combine between the environment and the production. For example, the combination between the solar energy and agriculture is one of the problem, one of the solution. Now we are implementing nowadays. For example, now the rooftop energy. Yeah, now some company they use a rooftop energy. Yeah, to use it to convert and to use in the agricultural activities there. It's one of the solution they are implementing nowadays. So I agree with you. So the problem is we have to find the good way to combine between the pollution, the environment, protect environment, and the produce agriculture. Yeah, and apply high tech in agriculture in Vietnam is very, very hard, very hard. That's why Vietnam is an agricultural country. Yeah, we are famous in the world. Now Vietnam is an agricultural country, not in the industry country. But the problem is the small scale is very popular. Yeah, and disseminate. And the second one is because the problem in the market, the problem in the price, the problem in the consumption is stop on the idea to invest in the long term. You know, for example, in Ho Chi Minh City, they plan this area in the agricultural area, but after 5 years, after 3 years, it becomes the residual area. Yeah. So it's the reason that the farmer they don't want to invest in the long term. if they invest a long term, if they have the time, they have to invest sustainability. But unfortunately, this is the problem now in Vietnam nowadays. It's not Ho Chi Minh City. I think it's also in the Mekong Delta area in the many provinces in Vietnam nowadays. Yeah. So besides that, many the reasons. Yeah. Stop. It can stuck to invest in high tech in agriculture in Ho Chi Minh City in the Mekong Delta area. So now I think with the Hien University and the Tho University, we are the scientists and we are the teacher. We have to teach the next generation. They have to think about the sustainability. They have to think about the benefit in the future, not the present time. Yeah. So I think it's one of the contribution we can contribute in the economic, in the social activities of Vietnam. Yeah. Thank you.
So next, Professor Wu.
Okay, the question is very difficult to answer because the term the best rice in the world. We not the proudest. We proud. We have the variety. Just got the best rice in the world, but the backside they have we call a hidden story about this, and like we confuse rice trademark and rice brand. The best rice in the world, not the rice brand. This the name after the International Rice Exhibition Fair in Philippines. Every year in the world they organize the fair like this. The previous fair they celebrate the Cambodian rice, and after that the Lộc Trời variety, and then Hồ Quang Cua variety ST25. But for rice breeding, just the second slide I show you, we are pay more attention to commercial rice breeding, not the basic knowledge like we apply any advanced in the technology in rice breeding. We are forgetting the basic research. We are forgetting the germplasm evaluation. We are forgetting about how to choose the best parents, how to select the promising progeny, and how to do the yield trial network. Yield trial network is very important in the whole country. This is many confusion. We proud this, but we need the time to text two things. The first, the variety ST25 just meet the demand of number one, distinctive, to be distinctive. It's different from another variety. To be uniform. Now, not uniform. Many phenotype, many. And the third one is must be sustainable from many environment and many years. So why today, Mr. Tung told me yesterday, it's not release a national variety. But we proud this, but we need the scientific thinking about this, because the pattern is not clear. And the selection processing procedure not clear. And then we celebrate Mr. Kua is my friend, very close friend. But we need the scientific step to evaluate the variety. Okay, and very easy. If you pay more attention to the background, you will have very good output. Thank you.
Yes, thank you, Professor Wu. So now, with Okayama University, I have two questions toward to the Okayama University. At first question, because Professor Goto, you say your major in horticulture and botanical science. I spent 2 years ago, I spent a day in your campus, even with the hospitals. And I had learned Okayama University very strong in fruit science and horticulture. So please update your research result. So I would like all the participants here to get more information, especially for the post-harvest technology in fruit. I know that you are very strong in proceed the technique to storage root like apple, peach, and send abroad. My second question, that's okay for the first question? First question about advanced technology in fruit research in Okayama University, especially to the post-harvest storage technology. So the first question. Second question, is okay for the second question? Second question relates to the graduate program in horticulture. I had learned from Professor Funahashi and Mayu-san about master and doctor program at Okayama University. At the last time, you had been done so well with Hien University. Therefore, if it's possible, can you summarize about that program? And I hope the young staff who sit in here, they can get more information and they can decide to apply to go to Okayama for their further study. Lastly, very personal, let's forward my congratulations to Professor Funahashi for the new position as the Vice President, and also say hello to Mayu-san. Thank you.
Oh, hi, I'm Goto. So my specialty is horticulture, especially floriculture, and I know the few post-harvest fact. So Okayama University post-harvest laboratory is very famous. So there are two laboratories for post-harvest. So they research the storage of grapes and peaches and persimmon, Japanese persimmon, and broccoli and kiwi fruits, especially fruit storage. So recently they found a good storage condition for peaches. Usually we keep 10 degrees for storage, but our groups found near 0 degrees is more suitable for storage. So maybe I have just a mistake, but maybe so 2 months or 3 months they or 3 months maybe. So if you have any question, please ask the post-harvest laboratory staff. Okay, first question is finished. Okay, so from now, second question, Dr. Yu introduce about the second question. Please, Dr. Yu. じゃあこっち来て。
Hello everyone. Hi, Dr. Yun. お久しぶりです。Okay, somehow my computer doesn't have the microphone, so I'm answering from using Professor Goto's computer. The question was, do we have the master's degree and then PhD degree especially for Vietnamese students and the young lecturers? We do have the program. One program is specifically for the Vietnamese students going to the master's degree. And then that's what we have been doing is with the Hue University. And we just welcomed the 13th batch students to our program. And unfortunately, because of COVID-19, we haven't been able to physically welcome them into Okayama University. But the basic idea is we provide this classes to those students, master degree students in environmental and life science. That includes the environmental science, animal science, and plant science. And those students are going to be admitted into our program, and then they will be spending 1.5 years in Hue University. And during that time, they will be learning about the subjects related to their study field. And then we also send our professors to Hue University so that they can give guidance. And at the same time, they will be learning a little bit of Japanese. And at the same time, they will be able to communicate with their potential supervisor. And after 1.5 years, they will be physically coming into Okayama University. And then, after they come to Okayama University, they will spend 1 year and taking classes, do the experiments, and then they will be finishing the program and getting the degree from Okayama University. That would be the one master's degree that we have. And then we also have the program for the young lecturers, especially from Vietnam. And we started this program a few years ago, and then we're targeting those young lecturers who are working in the university or organization or institute as a young lecturer or the researchers, and who are willing to go for PhD program. And the idea is we provide scholarship, and we call it sandwich program, and then we provide a scholarship to them, and then they will be coming to Okayama University and get the supervision for 1 year, first 1 year, and then they will also take classes. And then the second year, they will be going back to their own university or institute, and then they will be gathering a lot of data and then seeking for advice from their supervisor from Vietnam side. And then the third year, they will come back to Okayama University to start the data analyzing, and then they will be wrapping up their research and then write thesis, and then graduate with the degree, PhD degree from Okayama University. So every year we are accepting those applications for both master's and then PhD degree, especially the master's degree is fully funded, so they don't have to pay any while they're in Japan, and then they also get from us. Also, for the Vietnamese young lecturer who wants to go for PhD program, we do have funding, and currently we only have one spot, and then we just closed the application, but hopefully we will get a lot of applications from both Tho University and then Hien University, because this scholarship is only valid for those we have the MOU agreement with. So we're looking forward to receiving a lot of applications for next year. And we are very sorry that Professor Funahashi had to leave this conference a little bit earlier because he had another meeting. But I will surely send your regards to him, and then hopefully we will see all of you in Vietnam very soon. We are hoping to fly to Vietnam to meet all of you in near future. Thank you very much.
Ladies and gentlemen, we pretty much wrap up for the morning session. Now we're in a break for lunch at a number of restaurants. And I wish you to have a good lunch. And before you go, I would like to remind you about the afternoon agenda. Please come back here at 1 p.m. And we